To prepare total cellular DNA, the process begins with collecting a suitable biological sample, such as a bacterial culture grown under sterile laboratory conditions. The cultured cells are transferred into microcentrifuge tubes and centrifuged at high speed, allowing the cells to form a compact pellet at the bottom of the tube. Next, a lysis buffer is added to break open the cell membrane and release intracellular components, including the genetic material. To obtain cleaner DNA, enzymes such as proteinase K and RNase are used to remove contaminating proteins and RNA molecules. Cold ethanol or isopropanol is then added, causing the DNA to precipitate and become visible as thin white strands within the solution. The DNA pellet is carefully washed with ethanol to remove residual salts and impurities while preserving the purified genetic material. After drying, the purified DNA is resuspended in TE buffer or nucleus-free water, making it ready for downstream molecular analysis. The extracted total cell DNA can now be used for applications such as PCR, sequencing, cloning, and various genomic studies.